Thank you for joining us on the Neurodegenerative panel discussion. I am Suzanne LeBlanc, the Director of Clinical Relationships at the Focused Ultrasound Foundation. According to the Harvard Neurodiscovery Foundation, 5 million Americans now suffer from Alzheimer's disease, 1 million from Parkinson's, 400,000 from multiple sclerosis, 30,000 from amyotrophic lateral sclerosis, and 30,000 from Huntington's disease. Because neurodegenerative diseases strike primarily in the mid to late life, the incidence is expected to soar as the population ages. By 2030, as many as 1 in 5 Americans will be over the age of 65. Finding treatments and cures for neurodegenerative diseases is a goal of increasing urgency. This session will now begin with 4 separate pre-recorded lectures followed by a live discussion moderated by Dr. Jose Obeso. During the presentation, please feel free to submit any questions. We will collect these questions and answer as many as possible. It is now my pleasure to introduce the panelists that have provided pre-recorded PowerPoint presentations. Professor Jürgen Gutz is Foundation Chair of Dementia Research and Director for the Clem Jones Center for Aging Dementia Research at the Queensland Brain Institute. Dr. Nick Todd is an Assistant Professor of Radiology at Brigham and Women's Hospital with particular interest in focused ultrasound. He is especially interested in gene therapies crossing the blood-brain barrier for neurodegenerative diseases. Dr. Jose Obeso is a professor of neurology and the director of the Neuroscience Center in Madrid. He leads a highly active team to develop new treatments for Parkinson's disease and has led the field of focused ultrasound in this endeavor, publishing extensively in this field. Dr. Ying Meng is currently finishing her training as an MD PhD in neurosurgery at the University of Toronto. She has dedicated her thesis to focused ultrasound and is leading the next generation of clinicians to research this field. I'm sure you will find this session informative and engaging. Enjoy.
Thank you for the opportunity to join this session and talk about our early experiences with focused ultrasound modulation of the blood-brain barrier in patients with Alzheimer's. My name is Ying, and I work with Dr. Lipsman at Sunnybrook Hospital in Toronto. Alzheimer's is the most common cause of dementia. It is a neurodegenerative disorder that's characterized by abnormal accumulation of amyloid and tau in the brain. The search for disease-modifying therapies, which has focused on improving the clearance of these pathologic proteins, has been largely unsuccessful. This might be due to many reasons. It may be because we have the wrong target or the wrong drug, or simply because the drug is not reaching the target at sufficient concentrations. The vast majority of these therapies are biologic agents that have very limited blood-brain barrier penetration. Focused ultrasound, by transiently opening the blood-brain barrier, creates a window for the therapies to get through. The advantage of this technology over other potential drug delivery platforms, particularly for the Alzheimer's population, is the ability to deliver therapies non-invasively without open surgery to wide, large regions in the brain. And two, the advantage is that it is agnostic to the therapeutic of choice. The potential and promise of MR-guided focused ultrasound has been characterized in animal models by extensively by scientists in this session. In a pilot study, we looked to see whether this is feasible and safe in 5 patients with mild to moderate AD. Using MR-guided focused ultrasound, we were able to temporarily open a region in the right frontal lobe of these patients, and show by doing that, we showed increased gadolinium leakage, indicating increased blood-brain barrier permeability right after ultrasound. In repeating the scan the next day, we showed that the leakage is no longer there, indicating that the blood-brain barrier integrity has been restored. We also looked at amyloid PET scans, and the rationale is that, one, to look at whether blood-brain barrier opening can or will aggravate amyloid pathology, and 2, to see if some of the results seen in animal studies where blood-brain barrier alone can improve amyloid clearance, whether this holds true in human subjects. In the 5 patients we had, we did not find a group change, whether increase or decrease in amyloid tracer uptake in the area of cell notation. In a recent study by Dr. and his colleagues, they targeted the hippocampus, which is a key structure for memory and learning, and obviously very important for patients with Alzheimer's. They showed the technical feasibility and safety of opening the blood-brain barrier of the hippocampus temporarily. They also showed in 6 patients that after treatment, the amyloid tracer uptake in the treated hippocampus is decreased versus the untreated side. It's obviously very exciting, but what are the potential risks for patients with AD in these procedures? What we know so far is that there's been no serious procedure-related adverse events, and few adverse events have been reported. Community-acquired pneumonia that was not thought to be related to the procedure that led to the exclusion of a participant from subsequent treatments in the first study. 2 cases of transient MRI changes noticed in the patient after focused ultrasound that resolved basically the next day. So I will show you briefly these neuroimaging findings. Here on the left, we see areas of hypointensities in the T2 star change on the scan acquired immediately after focused ultrasound. These findings disappear or resolve when the scan was acquired the next day. We believe this most likely represents some hemosiderin or protein extravasation after blood-brain barrier opening that is cleared subsequently, explaining for this transient nature of the findings. On contrast enhanced flare, we see hyperintensities in the same pattern as the perivascular spaces that were observed in this particular patient on the T2 scans prior to focused ultrasound. And we believe that this represents a clearance of gadolinium that has extravasated across the blood-brain barrier as a result of increased permeability from the focused ultrasound through the perivascular spaces. These similar findings have been reported in a separate study in patients with ALS, where the focused ultrasound was targeted to the primary motor cortex to open the blood-brain barrier there. And here we have additional adverse events data to understand what patients are experiencing undergoing these procedures. This is a study of 4 patients, and we find that transient headaches or intraprocedural headaches are quite common, as well as discomforts related to the stereotactic frame and lying on the MRI bed. Notably, no late complications were reported in any of these studies. So, where are we now, and where are we going? So the long-term consequences of repeated blood-brain barrier opening and repeated procedures on Alzheimer's patients need to be better characterized. This needs to be studied at a much granular level, given the data in the subset of patients with Alzheimer's. The blood-brain barrier dysfunction has been linked to cognitive impairment. And the concern is that repeated blood-brain barrier modulation over time will have harmful effects on patients with Alzheimer's. What is the significance of the T2 star signal change in patients? We will learn more as we have more patients undergo the procedure that have these signal changes. Is the targeting of multiple widespread structures in the brain feasible with focused ultrasound and MR-guided focused ultrasound specifically? Can the workflow be optimized to be faster and less disruptive on the patient's daily functioning? We are trying to address these questions and more through a current study underway at Sunnybrook using advanced neuroimaging and biological markers of blood-brain barrier integrity in Alzheimer's disease pathology. Here, we aim to have 30 patients enrolled in the study, and each patient will undergo focused ultrasound procedure 3 times over the course of 6 weeks, and we'll be targeting areas of the default mode network, which harbors amyloid pathology early, and specifically, we will be targeting the hippocampus, bilateral anterior cingulate cortex, and precuneus. Thank you for your time. Again, I'd like to acknowledge our collaborators, as well as foundations that have supported us along the way.
Hello, my name is Jürgen Götz. I'm based at the Queensland Brain Institute in Queensland, Australia. And I would like to thank the organizers of the 7th International Symposium on Focused Ultrasound for inviting me to join this panel. So I would like to talk about opportunities and challenges of treating Alzheimer's disease with therapeutic ultrasound. There are many types of dementias. Alzheimer's disease is the most important one, but we appreciate by especially looking at the long list of non-Alzheimer's disease dementias that in all likelihood each dementia subtype will require a specific treatment. This also suggests that the clinical benefits will vary between Alzheimer patients and that more than one therapy will be required even with the Alzheimer type of dementia. Focusing on Alzheimer's disease, there are two key players, A-beta and tau. A-beta forms plaques, tau forms tangles. There's also genetic evidence with familial cases being identified with mutations in a gene which is linked to A-beta production. And there is a subset of familial frontotemporal dementia where mutations have been identified in the gene which encodes tau. So together, there's histopathological and genetic evidence pointing at A-beta and tau, and my lab operates on the assumption, leaving your inflammation aside, that A-beta and tau initiate and drive the degenerative process that characterizes Alzheimer's disease. We have found in our work that A-beta being extracellular is an easier target for therapeutic ultrasound. Tau being intraneuronal is a much harder target, most likely because not only the blood-brain barrier has to be overcome, but also the plasma membrane to reach tau in neurons. This slide summarizes some of the work we have done in the Alzheimer's space. We have shown that therapeutic ultrasound on its own with microbubbles is able to remove amyloid and restore memory functions in Alzheimer mice. We used other Alzheimer mouse models showing that tau can be partially removed, memory and motor functions can be restored. And we also found that when we use a combination therapy using an anti-amyloid or an anti-tau antibody, that these combination therapies are more effective in removing amyloid and or tau and in restoring memory functions. The challenges which we encounter in treating Alzheimer's disease are very generic. They apply to all neurological diseases. There's an incomplete understanding of the etiology and progression of the disease. Importantly, a lack of robust and sensitive biomarkers, because it's obviously critical when we want to treat early, when we have these biomarkers, and it's also required to validate the efficacy of a treatment. This slide is critical because it makes a point that A-beta and tau pathology spread through the brain. We do this slowly. They start in particular brain areas, but they're also found in different brain areas. And again, it differs from the pathology we find for alpha-synuclein. And what this indicates is that the earlier we treat, the smaller the brain area is we need to target, but also that when we treat early, in order to remove A-beta and tau, we need to treat different brain areas. Another challenge is that it's not completely clear what the actual toxic species is. Tau and A-beta come in different isoforms and different variants. They go through an aggregation process. They undergo post-translation modifications. It's massive. Tau has 85 phosphorylation sites. Do we want to remove A-beta and tau more generally, or do we need to target specific species? I would like to finish on a positive note. When we look at all dementias, all neurodegenerative diseases, they are all characterized by one unifying feature, and that is protein aggregation. Having shown that A-beta and tau can be removed by therapeutic ultrasound, I'm convinced that all these other protein aggregates can be similarly removed. I would like to finish by stating that I believe that the opportunities of using therapeutic ultrasound as a treatment modality for brain diseases by far outweigh the challenges. Thank you.
Oh, hello. So this is a summary about the role of focused ultrasound in the treatment of neurodegenerative disorders. And clearly, this has a dual approach, symptomatic ablative treatments, which essentially are only available for Parkinson's disease to improve the cardinal motor features of Parkinson's disease, and blood barrier opening, which is mainly intending to be used to deliver for the delivery of putative neurorestorative protective agents. And this is an ample area of neuroscience. And we would have to think about who should be treated or could be treated, when to do that, and how to treat it. And my main message today is that whatever we do, we should do it early. There's no point in waiting for the disease to be evolved, because the chances to interact in a positive way to stop and really improve the process is much lower. So these are the major neurodegenerative disorders that everybody knows. Sorry about the Spanish: Alzheimer's disease, Parkinson's disease, and ALS. And they have different features, like in Alzheimer's disease is tau protein and the amyloid deposit, the ones which are featuring the disease, whereas in Parkinson's disease is the inclusions known as Lewy bodies, synuclein positive, and ALS. There is also tau, but this is affecting mainly the corticospinal tract and then later on the front. And they all progress from relatively focal, like in Alzheimer's, in the hippocampus, to the neocortex, and eventually extending to many other brain regions, and similarly in Parkinson's disease. So they have in common to be relatively focal at onset and then spread around the central nervous system to become quite systematized. Another important disease, thinking in who could be treated first, is Huntington's disease. And the important point with Huntington's disease, of course, is that the genetics of Huntington's disease is very well known, and the pathology is also well known, affecting mainly the striatum first, and then the neocortex, particularly the prefrontal and parietal cortex. And most importantly, there is the possibility of being really early diagnosis of Huntington's disease, like when it is completely in a prodromal, even a pre-symptomatic period of evolution. Therefore, the disease could be attacked, could be treated well before the neurodegeneration is extending and becoming really too general. There are a number of studies, particularly experimental, with several very modern approaches to treat Huntington's disease. And this is one, the ASA, the antisense oligonucleotide, which is promising, although it's still not completely clear how much it could really enter into the targets like the striatum or the neocortex. So focal delivery very early in the evolution of Huntington's disease by VBB opening is a real option. Same for viral vector genetic expression, which currently is being tried using surgery, and it's not a minor procedure to do bilateral surgery to deliver a vector in the striatum. So I really think that BBB opening has a real role here in the near future. My work is mainly on Parkinson's disease. Parkinson's disease at the beginning is really focal in the substantia nigra, starting by the ventrolateral region and producing dopamine depletion in the posterior putamen, as you can see here in the F-DOPA PET, very limited to the posterior putamen. Early in the evolution, Parkinson's disease is really focal and pretty restricted to certainly to one body parts and typically to one or two adjacent body segments, like you can see here for the shoulder. On the arm, the patient has a very good face expression, good walking, but is particularly the shoulder which is affected, as you can see in this typical maneuver. So in those type of patient, Jose Pineda in my team and a number of colleagues have done a study showing that early in PD evolution, F-DOPA PET shows a particular pattern which is important uptake depletion in the posterior putamen, as seen in this axial PET scan, very asymmetrical, the most affected site, and mild in the asymptomatic side. Doing cluster analysis, that depletion in the sagittal plane can be seen here, and somatotopically in the body, corresponding to the interface between the lower limb representation and the upper limb representation, more or less where one could envisage the shoulder indeed to be placed. So we have a fairly good representation of where dopamine depletion in the nigrostriatal pathway begins in Parkinson's disease, and I think this is a perfect example for trying to intervene at this point to act against the progression of Parkinson's disease. Parkinson's disease also progresses in a proportion of patients, relatively high proportion of patients, to PD dementia, and this is more a cortical phenomenon, although not only. And here I'm following studies from Toronto, mainly doing BBB opening in the cortex in Alzheimer's disease, and in ALS patient we have done 5 patients, 10 treatments to do opening of the parieto-temporal cortex, occipital cortex, as you can see, the gadolinium enhancement here, and moving away, disappearing, fading, 24 hours, same in the coronal, etc. In most of the patients and sessions, this was achieved at this extent. And we have moved to the posterior putamen now, mainly because this is our real target for early intervention, and we're achieving opening in this region too, although not as robust yet as we would want. So I think neurodegenerative disorders like Huntington's and Parkinson's can be are very good, a very good type of diseases to be considered first for neurorestorative protective therapies by BBB delivery opening. And the real key is to detect these patients and to treat them really early. And of course, we have the problem of what could be the best of the available treatment possibilities to start as soon as possible such a pilot studies. Thank you very much.
Good morning. My name is Nick Todd, and I work in the focused ultrasound laboratory at Brigham and Women's Hospital. When I was invited to participate in this panel on neurodegeneration, I was asked to come up with a question as a burning question for the field. As most of my work is in focused ultrasound for drug delivery to the brain, I'm interested in the question of whether FUS-BBB opening can enable IV administration of gene therapies. So in this short presentation, I'd like to try to put this question into some context, considering both how gene therapies are typically administered now in clinical trials and what are the challenges that focused ultrasound needs to overcome before it can contribute to this clinical successfulness. So in the current landscape of how gene therapies are delivered to the central nervous system, the general trade-off is between intravascular injection, which is safe and repeatable, but has a difficult time in delivering the therapy across the BBB to reach the brain, against more invasive procedures that bypass the BBB by doing a direct injection into the CNS, such as intrathecal or intraparenchymal injections, have essentially the opposite trade-off. So currently, all different routes of administration are being used in a number of clinical trials. You can see this table here is from a recent review, just mentioning some of the diseases and therapies that are being given, and they're being given intrathecally, intravenously, and intraparenchymally. And in fact, the first FDA approval for gene therapy directed to the central nervous system was for an AAV9-based approach that was given by intravenous injection. This was for the disease of spinal muscular atrophy. But two things to note is that these are for children who are 2 years of age or under, and the target was motor neuron cells, which reside primarily in the spinal cord. So this may be an easier target than delivering gene therapies to an adult brain. But probably the biggest hurdle right now for IV delivery is that at high doses, particularly for AAV-based delivery vehicles, there's a limit above which you can't go. And that can be seen here in a recent trial that had to be halted due to toxicity and an inflammatory response in patients who died in the high dose arm of this gene therapy trial. So these more direct methods are still being pursued to overcome this problem of delivery. And here you can see an example of an intraparenchymal injection for Parkinson's gene therapy that's being delivered. And you can see you can reach deep brain targets and get sufficient concentration, but perhaps the limitations are about how much coverage you can achieve. And then this MRI shows you roughly the area of distribution that you can get. And then finally, in the current landscape, well, they haven't reached the clinic yet. These self-crossing AAV capsids are coming. So this is a figure from a 2016 paper showing a new engineered capsid compared to a traditional AAV9 vector. And you can see at the low dose, and in particular at the high dose, you get a much greater penetrance across the BBB and into the brain. Although it should be noted that there are still limitations. So these right now appear to be species specific, and that, well, it might work in one animal, it doesn't work very well in another animal, and to my knowledge, they've not yet been tested in humans. So given this landscape and this question of can FUS-BBB opening be used to try to overcome these challenges, what are some of the more specific questions that we need to address? So I think that probably the primary most important one is if we use FUS-BBB opening, will that allow us to reduce the systemic dose level while still achieving a good enough concentration of the therapy into the brain to have a therapeutic effect? And then other questions include, can we use BBB opening to get better coverage throughout the brain for a lot of these neurodegenerative disorders or whole brain disorders? And one of the main advantages of focused ultrasound BBB opening compared to the neurosurgical injection approaches is its repeatability. And so can this be leveraged in various ways? For example, to achieve that better coverage, but then we have to take into account the immune response that the body has to AAVs for those types of approaches. Well, what about for approaches that need perpetual dosing? Can we continually disrupt the BBB to deliver, and so on? And then also, once we've disrupted the BBB and get the therapy in, can we optimize the procedure to let the gene therapy uptake in neuronal cells where it's typically needed the most and not get swallowed up by astrocytes? And then finally, as these self-crossing AAV capsids get better and better, can we somehow combine the two technologies together to make a really efficient and efficacious approach? So in closing, I'd say that a lot of the trade-offs that are faced are both disease and therapy specific. And I am generally optimistic that ultrasound BBB opening can in fact find a kind of home run application similar to what was found for ablation and essential tremor treatments that will allow the field to have a great clinical success and then move on to further applications. Thank you.
Hi, everybody, and welcome to the Neuro. I'm Suzanne LeBlanc, a representative of the Focused Ultrasound Foundation, and I'll be filling in as the moderator until Dr. Jose Obeso returns from another session that he's just finishing up. We're privileged to have Dr. Nick Todd, Dr. Jürgen Getz, and Dr. Ying Ming with us to have this discussion. Please feel free to submit any questions you have in the live Q&A or chat functions, and we'll try to get to them. But I'm very excited about having this discussion. There's been a tremendous amount of research over the last few years that have really advanced the use of focused ultrasound in this field. So I'd like to get started. And Dr. Todd, perhaps you can get us started with your exciting work on gene vectors and the research you're doing and where you think it's going to lead to. Go ahead.
Sure. Thanks for the invitation and the opportunity. So our interest is in using focused ultrasound BBB opening to deliver gene therapies to the brain. And there's a lot of exciting work, as we've seen recently, on Parkinson's and Alzheimer's disease, but we're actually looking at some of the more rare single gene disorders. So for example, Huntington's disease, or some of the lysosomal storage disorders that have a neurological manifestation. As we think that these could be good candidates for application because they have a clear target of this single gene mutation. And also in the preclinical models, they have a clear readout. If you can deliver something that would lower expression, then you can sort of clearly demonstrate success. So we're trying to get 2 programs going for Huntington's disease, as I mentioned, where we're delivering an AAV9 vector, I think is what we'll focus on. And then also lysosomal storage disorders. And so we're hopeful that these will show good results. We're in the early stages of showing delivery efficacy, and now we want to move to try to demonstrate a therapeutic efficacy in these preclinical models.
Right. I think we can make some comments or questions specifically so to try to put this into some action. So, Nick, I agree entirely with you that, particularly monogenic genetic disorders, particularly Huntington's is a very good example because the gene is known, the protein is known, a lot about the mechanism of action of the toxicity of Huntington is known. And so the disease can be diagnosed quite earlier, which I think is a fundamental feature for any neurodegeneration to pick it up really early whenever it's possible. Now, my question to you, Nick, is how do you know what you're thinking to deliver to the striatum and or the cortex will do the job?
Yeah, I think that's the big question. For Huntington's, we'll start with the striatum because we know that's where the disease manifests initially, but we also know that it's a whole brain disease, and likely a lot of the later deficits come from cortical degeneration. And so we'll, to fully treat the disease, we probably will have to hit cortex as well. And so the problem of not just delivery efficacy in one region, but being able to target multiple brain regions, I think is a big challenge.
Yeah. And are there plans, advanced plans to start doing this in patients? I mean, in carriers, I would say, I guess, or early patients.
Yes, so not for us yet. We're still looking at mice, and our next step would be non-human primates, and showing safety and delivery efficacy in non-human primates. And then at that point, then maybe we could be ready to go to a trial, but we're not at that point yet. Very good.
So, Jürgen, do you want to go on and give us a little summary?
Yeah, absolutely. Thank you. I have a long-standing interest in Alzheimer's disease, and the field has for a long time been dominated by amyloid. So we have the amyloid pathology and the tau pathology. And historically, clinical trials have focused on amyloid, and more tau is entering the focus. And I think most of you know that there's kind of this debate whether aducanumab should be approved or not. Aducanumab is an anti-A-beta antibody. But we found, and the big question in the field is what is actually driving disease? Is it amyloid or tau, or is it something else? So when you look at the clinical trials, also in the focused ultrasound space, the question is, what do we want to remove? Do we want to remove amyloid, or do we also want to remove tau? And in my own research, we have animal models with an amyloid and with a tau pathology. And it turns out that tau is a much harder target. And this somehow makes sense because amyloid is extracellular, so in a sense it's more accessible, and tau is intraneuronal, so it's much harder to access. And in a sense, in order to remove tau, which is intraneuronal, one needs to overcome the blood-brain barrier, but one also needs to overcome the plasma membrane. And then the question is, we work with antibodies, so we develop our own anti-tau antibodies. And the question then is, does the antibody need to engage with the target? And even when we look at classical approaches where immunotherapies are being pursued, one will find in most studies that the groups don't show that an anti-tau antibody actually engages with tau, with intraneuronal tau. And then it's also not understood how tau is then being cleared once the antibody has engaged. So I think there are a lot of questions. And when I was asked to come up with a burning question for this panel, I thought one question would be, for example, how do we deal with differences in the uptake of therapeutic agents by different brain areas or cell types? What we find in our own research is when we look at amyloid and tau that, for example, you can clear amyloid, and Lanenka has presented this, in the cortex, all over the cortex and in the hippocampus. When we look at tau clearance and the uptake of anti-tau antibodies, we find that this is often confined to certain brain areas. For example, a mouse model with an amyloid, with a tau pathology in the amygdala and in the hippocampus, but it's much easier for us to visualize a fluorescently labeled anti-tau antibody after delivery with focused ultrasound in the hippocampus as compared to the amygdala. The question is, why is that?
Right. Anybody can?
Jürgen, do you think it's a delivery question of you're not opening the BBB as much in these areas, or an uptake question of the
uptake
drug is in but not getting uptake?
I think it's uptake and retention. I think it's not only uptake, but whatever we are delivering therapeutically also needs to be retained. And it needs to engage with the target, and we need to ensure that it's not being cleared. It's not related. I think that's a separate problem one is dealing with, differential blood-brain barrier opening. I think all of us see that. But another issue is even with a consistent blood-brain barrier opening or comparable blood-brain barrier opening, that there are differences in therapeutic antibody uptake.
Yeah, that may well be. Jürgen, you mentioned just a minute ago about the amyloid and tauist and all that, and obviously this is bound, to be a combination. But my worry at this is very early stage of BBB opening and therapeutics is that, there is abundant, like nearly 30 or so studies with trying to reduce amyloid from the brain of Alzheimer's patients, which has failed.
I think the argument which has always been brought up that one is treating too late with too little. And in a sense, when you look at the aducanumab trial and compare the two phase 3 trials, when one does a post hoc analysis and looks at the total cumulative dose of the antibody, one finds clinical benefits for those patients where the highest dose has been delivered over the treatment period. And this is kind of also reflected by our animal studies, where we find that when we use aducanumab together with ultrasound, we can increase the dose in the brain, and hence we see better functional outcomes when we look at the active place avoidance test. I think in the clinical setting, the issue is simply that one is not delivering enough antibody. And I think that's something where ultrasound can help, because it allows firstly to operate with lower doses. So an issue such as ARIA may be preventable. And it's also possible to combine two antibodies without reaching levels which would be toxic.
Right. two questions here. One is, which is, I guess, for you, Jürgen. If you could explain a little bit better, how can just pure fast BBB remove some plaques or tau from the brain that I can qualify in the mice so far?
Yeah, I mean, what we found, not only we, but also others, is that by using ultrasound on its own without a therapeutic agent, but by using microbubbles for blood-brain barrier opening, that glia are activated by the uptake of blood-borne factors, which are unidentified. So microglia are activated and they take up the amyloid into their lysosome, where amyloid is being degraded. And for tau, we have shown that ultrasound on its own with microbubbles activates neuronal autophagy. I think that's the beauty of focused ultrasound, that different mechanisms are activated, so one can clear amyloid and tau by using both glial and neuronal mechanisms.
That seems too good to be perfectly right. And you could any lump in proteins. And then, anyway, let's ask Jean Mann. Please, can you just give us a little insight into your work?
Thank you, Dr. Rubito. So I'm from Sunnybrook, and I guess I take more of a technical view on this portion of the seminar. So my work has been looking at whether we can scale up this technology to treat multiple areas in the brain. So, neurodegenerative disorders such as Alzheimer's is a disease disorder. So we began with our phase 1 study looking at the right frontal lobe, and now we are looking at hippocampus and regions of the default mode network. We're also really interested in how we can deliver the ultrasound safely. Different energy powers we can achieve different extent of BB opening. So how do we deliver ultrasound with adequate feedback such that we can open the blood-brain barrier just to the extent that we want it to deliver the therapeutic without causing hemorrhage and things like that is something we are interested in looking at. And also how BB opening in widespread regions of the brain is changing biological markers is something we're also very much interested in looking at in our studies.
Right. And do you foresee everything being just local, or could there be a chain reaction, let's say a succession of events? If you modify signaling, right, you could, and that's very critical for patients, because let's suppose Nick promptly treats early HD, Huntington's. It would be wonderful if one could modify the sequence of events, right, rather than get later to try to battle with the whole brain deteriorating. What do you think? Is that possible?
I think it certainly is possible. I think I think the effects of focused ultrasound is not just locally. We're seeing changes on flare signals that suggest that we're changing the flux of lymphatic pathways. We're also looking at biomarkers that are part of the BUG integrity, like S100 beta. We're also looking at markers of axonal health, like NFL, just to see how things are changing in a more holistic systemic level. And we're noticing some changes there as well. So I think what is happening with focused ultrasound is not confined to a single location, and it probably has more widespread effects than we currently have data for right now.
Okay. We have a question for the Nick Todd here. The question is, do you expect AAVs to provide similar transduction patterns after fast compared to direct injections? That's a good question.
So I think it's clear we'll get far less concentration of the AAV compared to a direct injection. So the question to me is, can we get enough in to have a therapeutic effect and clear that bar? And then the next advantage of using FOOS would be, compared to direct injection, it's more or less a one-time procedure, and you can choose your one brain region that you want to hit. So then can we use the repeatability of FUS, because it's much less invasive and safer, to get that enough concentration in, but at multiple brain regions? And I think it's clearly the answer is yes for a lot of therapies, but as was alluded to earlier, the challenge of AAVs, I think Dr. Caplet brought this up in a previous session, is that there's a host immune response. And if you give one dose targeted to one area, if you go back a week, 2 weeks later to try to deliver to another area, you may not be as successful because of this immune response. And so I think for gene therapies, that's going to be a challenge of how do you get this kind of whole brain coverage that you might need for a lot of these diseases.
Right. So we got 2 minutes for closing. So I need brevity here. I was going to ask anyone, there's a lot of emphasis on with viral vector delivery, but in the Parkinson's area, much more is going on with antibodies. And antibody has been, there are 2 good studies in PD, very early PD, and the antibody is given IV monthly. So monthly may be a bit too much for, meanwhile, the procedure requires a frame. Maybe that when we go frameless, may be different. But do you foresee the possibility of using not genes, not gene therapy, but molecules, and do it repeatedly? You need to be brief, sorry.
I think hopefully yes, but it remains to be seen.
I mean,
I think a lot of...
Jean?
Sorry, I was just going to say, I think a lot of technical modifications need to be changed. As you said, the frame and the hair shape, those kind of things need to improve for patient tolerability to have agree to go through monthly treatment forever for the rest of their life, essentially.
Thank you. Jürgen, very quickly.
Yeah, I don't think it most likely needs only a few treatments. I mean, we find this with the amyloid and tau pathology. I mean, 4 treatments are sufficient, and most likely even with a virus or an antibody, maybe a few treatments are sufficient.
Very good. Upon closing, I'd like to mention to the audience, well, thank everybody, but to the audience that several of these diseases have pathogenetic, the pathophysiology involved not just the very causative mechanism, but also the underlying symptomatic mechanism. And approaching PD is a perfect example. One could approach PD both symptomatically and etiologic through focused ultrasound. And I think that could apply eventually also to Huntington's and ALS, etc. Alzheimer's is a bit more difficult to envisage, but that we'll be hearing about that in the future. Thank you very much, you all, and those back there who we don't see, and the foundation for running effortlessly this virtual meeting. Bye-bye.
Yeah, thank you.